Welcome, everyone, to today's webinar about the applications of COVID-19 diagnostics and key unanswered questions in low-resource settings. The webinar was organized by the Virology, Immunology, and Diagnostics Working Group of the COVID-19 Clinical Research Coalition. The coalition was put together early on during the pandemic in order to facilitate and fast-track clinical research in low and middle income countries. You can find more information about our working group on the coalition's website. We will post a link to our working group's webpage in the chat. We are looking to expand our working group's membership. So if you would like to join us, you'll also find the link on the webpage and again in the chat here. I am Chris Woods. I'm a professor at Duke University in the departments of medicine and pathology here in the US, and co-chair of the working group, along with Dr. Shaima Inani, a professor of microbiology from the Suez Canal University in Egypt, who is also joining us today. I am delighted that we will be featuring two diagnostic experts from the working group for today's webinar. Their expertise ranges from the highly technical to the highly practical. Following their panel presentations, we will have a 15-minute question and answer session by Professor Inani. Just a few housekeeping details. Today's webinar is being recorded and will be posted later on the coalition website. Please ask your questions via the Q&A tool at the bottom of your screen. You can also upvote people's questions by clicking the thumbs up icon, which will send those questions higher up the list to prioritize during the question and answer session. If you would like to address a question to one of our speakers in particular, please be sure to include the speaker's name with your question. So before I introduce our first question, we would like to know a little bit more about you. So we are going to post a question on your screen, which you can answer directly, which you should be able to see now. We will share the results with you in a few minutes. All polls are completely anonymous. I'll give you 15 to 20 seconds to answer the questions. Okay, why don't we review our distribution? All right, so we do have a wonderful distribution here, a large number of individuals who are participating already in the COVID response and involved diversely through clinical research, diagnostic research, clinical care, and laboratory diagnosis. Thank you very much. One of the major work streams of our group has been to generate key research questions relevant to the goals of the work group. They are listed here. We have highlighted areas for study that are related to self-collection, to both the utilization of serological and antigen-based testing, assessments of transmissibility using newer diagnostics, and emphasizing the need for biobanking to support these and other questions. So, the first talk today will be provided by Professor Wolfgang Preiser. Dr. Preiser is a medical virologist whose principal research interests include emerging and potentially zoonotic viral diseases. He served as an advisor to the World Health Organization on the severe acute respiratory syndrome in China in 2003, and led the Frankfurt group, who together with colleagues from Hamburg were among the first to isolate the SARS-associated coronavirus in 2003. Since 2005, Dr. Preiser has been professor and head of the Division of Medical Virology and in the Faculty of Medicine and Health Sciences at the University of Stellenbosch National Health Laboratory Service in Tygerberg, Cape Town, South Africa. Dr. Preiser.
Thank you very much, Chris, and good afternoon, good morning to the participants. Thank you for giving me the opportunity to give my presentation. Just in terms of my background, this is our faculty. In the background, you see Tygerberg Hospital. We are, as virology departments, oftentimes are on the top floor of the building. I don't know why that seems to be a tradition. And we have a dual purpose. We are both the academic unit of the Stellenbosch University doing research and training and teaching on virology. But at the same time, we are part of the South African National Health Laboratory Service that provides diagnostic testing for the approximately 80% of the South African population that do not have private health care. And our role is providing all virological testing for approximately half the population of our province, the Western Cape. So it's a large number of patients whom we serve. Yeah. The gold standard for diagnosing COVID-19 is detecting the nucleic acid or genome, RNA genome of the causative virus, SARS-CoV-2, from a specimen, typically and most commonly used nasopharyngeal swab specimen, which is then eluted in the lab. Viral RNA is extracted from the specimen, subjected to reverse transcription, and then amplified by real-time PCR, giving a result in these typical curves. And this is a widely used graph from a JAMA paper a while ago, and it shows the course of COVID and the infection. And you see this is on the top bar. I have indicated where PCR on a nasopharyngeal swab is usually positive, and that starts about 2 days prior to onset of symptoms, importantly, so people are also potentially infectious prior to becoming symptomatic and thereby recognizable clinically. And then it lasts for a variable period of time. And as real-time PCR gives a semi-quantitative signal, at least one can sort of assess the viral load in these specimens, and it declines. And we know that patients are usually infectious from 2 days prior to up to 1 week after onset of symptoms, and beyond 10 days, there might still be and often is detectable viral RNA, but usually no infectious virus anymore. And then beyond that, we also know from our own experience that many patients actually shed viral RNA for prolonged periods of time, sometimes many weeks, and I come to that later on in my presentation. And this is the only thing that I want to say about antibody testing, because we have an excellent talk on the topic coming up later on. But obviously, given the biological and other constraints, it's not the method of choice for diagnosing current COVID-19. We now start with the first poll. I'm not going to read it, but I would like you to use the next 10 to 15 seconds to quickly go through and just tick what you think applies. So tick everything, each one of the 5 statements that you think are true. As Chris has said, this is all anonymous, so you won't be marked on that. So let's have a look what our participants voted. Right. So sophisticated lab techniques need skilled staff and expensive equipment. Indeed. Testing of swab samples can easily be automated. I would dispute that. The infection risk is unfortunately a factor, and therefore those who are obtaining these samples need to be wearing PPE. And absolutely true, neither are reagents and consumables widely available. And again, I'm going to go into this in more detail later on. And antibody testing, I think I've given that away already in the previous slide, is not the preferable method, even though it, of course, it can be done given using suitable tests at point of care. Thank you. Now let's move on. So one has to say that I'm not aware of any other newly emerging viral infection where the causative agent had been discovered, sequenced, and assays been developed. And development in this case means in silico. So they had been developed given using the available sequence information and, of course, drawing on experience with related viruses from previous occasions. And all that happened within a few weeks, which is absolutely marvelous and also points to the power of modern science and of people being linked to one another in virtually real time. So that's an amazing achievement, but that is not where it all ends. The enormous challenges were for numerous, many thousands of diagnostic labs worldwide to establish and validate and finally ramp up from literally zero to many thousands, tens of thousands of samples a week, these new diagnostic tests within weeks. We have realized that the sample type plays an important role, as does sample quality, nasopharyngeal swabs versus nasal swabs versus saliva versus stool. And of course, uh, they all have their advantages and disadvantages. At this stage, nasopharyngeal swabs remain the gold standard, even though particularly nasal swabs and saliva have advantages, often because they can be obtained by the patients themselves. And there are good studies to show that one loses some sensitivity, but the advantages may outweigh that problem. Swab samples need preparation. We call that prepping in the lab. It's not it's not something which you can just slot in a label tube into the a machine that then does it from there. It's something where you need an operator, and of course they are potentially infectious, so need to be handled under appropriate biosafety conditions. PCR as such is a demanding technique still, even 30 years after it was introduced in diagnostics. It requires skilled staff and special equipment. And we also then ran into issues that we've never encountered before, shortages of various reagents and consumables. It started with swabs that suddenly, I mean, seems simple enough, but suddenly they became scarce worldwide, and people were trying all sorts of alternatives to make do. Buffers ran out. They even threatened to be a global shortage of ethanol. Can one believe it? Which is important for RNA extraction. So we had to contend with all sorts of issues that we had really not been expecting. And I think where the world has woken up to realities that we were not aware of before. And being working in a resource limited setting, I must say that I take some consolation from the fact that these struggles are not limited to us. They also affect many or most industrialized countries in their settings. Of course, solutions come to mind. One would be high throughput automated assays, and they do exist. Um, of course, one needs to have the equipment, which is typically hundreds of thousands of dollars worth of equipment. It's highly sophisticated, needs an appropriate setting, needs the skilled staff. But then when you have it, and there are labs, of course, even in non-industrialized countries that have them, particularly for HIV viral load testing, you still need the kits. And to the present day, there is still a global shortage of these kits for the few fully automated systems that are available. At the other end, one has the point of care or at least near patient systems, and below are pictures of a widely used system, DNexpert, which is used in many developing countries for TB diagnostics. Again, that's perfect. It's a state-of-the-art molecular technique. It runs largely automated, and it can be done close to the patient. So basically, smaller labs in smaller laboratories can run this. Big problem is and remains again to the present date that there's a global kit shortage. So we don't get enough of these kits to allow this testing. So we were facing then in April, May, and June, and I very much fear that here in South Africa, we are now going to face a similar situation once again. An incredible number of samples. This is just, this is the amount of samples that came in several times a day over. And that met and is possibly meeting again a dire shortage of test kits, reagents, and consumables. So what do labs do to work around these issues? You need people, train additional staff and volunteers, and that's something we've done. And it was absolutely amazing to see how people came forward from other departments, other laboratories, but also from outside, trying to help. Of course, they will not be able to replace highly skilled and trained and experienced molecular scientists and technologists, but they can take over certain other aspects of the workflow and still make a huge difference. We realized that we had to optimize referral logistics and internal workflows, and we did so, and it made a big difference. Many labs worked around certain shortages, in this particular case of extraction reagents, by introducing simplified procedures whereby one does not formally extract viral RNA from the sample, but just performs a lysis and a heating step, which works generally well, but has its own caveats and is not, is not a very simple quick fix. And one has to be careful to preserve the sensitivity of testing. Pool testing is something that definitely works. Problem being that it can only be used where you have a very low pre-test probability of having positives. Otherwise, the pools of samples, it basically means testing several samples in one test together. And if the positivity rate shoots up as it is doing here again now, all pools will be positive, so nothing will be safe by using pool testing. What we find very important is diversification. Unless one is able to mix and match between different systems and kits and suppliers, one will run out of one or the other at some stage. The downside is, of course, that this cannot be done by people who just know on how to operate one automated system. It requires well-trained staff, sophisticated equipment, facilities. So it's not a simple solution, again, that would suit all settings automatically. And that is one of the systems that we were fortunate enough to receive during the outbreak. And that, of course, helps a great deal. But again, it depends on certain supplies in terms of plastic consumables and reagents, and there is limited flexibility. So my advice to anybody would be as flexible and open in terms of systems as you can be. This is for molecular testing, which remains the gold standard. Now, seeing that many countries, and that's not just developing countries, are struggling to test enough, and we can tell when the numbers of test requests go up, but also the positivity rate. It basically tells us we are not testing enough. And that is the same that has been experienced in many places. Some countries have never quite been able to overcome that problem. Others did better in between, but are now with a renewed surge being overwhelmed once again. So rapid antigen tests seem a promising alternative to nucleic acid testing. Most of these assays detect the SARS-CoV-2 nuclear protein. They provide a result very quickly, up to 15 minutes, and then you have it. So it's also very advantageous. Can be used at point of care near patient. They are generally very simple to do. So nurses, for example, and clinical doctors can be trained to do them. They're relatively inexpensive as compared to nucleic acid testing, but they have downsides, of course. And the main thing is that they are generally less sensitive than nucleic acid testing. The hope being that they would still be able to identify most people who are currently infectious. Now, this is another graph that shows here negative tests are the empty little circles, and the filled circles are the positive tests. You see the viral load curve, and it's basically the same course of acute SARS-CoV-2 infection that you've seen before. You see the viral load as measured by real-time PCR going up, peaking, and then declining over the weeks post onset of symptoms. And you see in the middle and indicated by the horizontal line, the dashed line, what a rapid antigen test with low analytical sensitivity would be expected to catch, to diagnose. And you see that there will be a period prior to the onset of symptoms where PCR is sensitive enough to identify infection, but an antigen test probably not. And then subsequent to onset of illness, we expect an antigen test to be able to identify patients for about a week and not longer than that, whereas PCR should be able to detect them as positive still 2 to 3 and sometimes many weeks later. And remember that this is also the time period when people will fall seriously ill and need hospitalization and therefore need to be diagnosed. And now let's have the next poll, and that is about antigen testing. Again, I'm not going to read it. I'm just going to give you 15 seconds to look at this. Which of the following are true about rapid antigen tests for diagnosing COVID-19? Right, shall we look at the results? So I would fully concur with the first answer, allow widespread testing, is simple to do and cheap. Yes, I don't think it is an advantage that they are of lower sensitivity. Um, I also concur with the third answer, even though confirmation by PCR testing may be needed for many, but possibly not all applications, that one can do it very rapidly and everywhere basically is still valuable in many cases. Um, Also based on, and we don't have a lot of experience yet with the rapid antigen test for COVID, but we know from rapid tests for other entities like HIV antibody tests, we know that operator error is immensely important. So if people are not well trained and constantly supervised doing them, they may do them wrong and the results may be unreliable. And of course, antibody testing, yes, it can be done on finger prick blood, but of course, it's not an alternative as we've said before. So thank you very much for giving your opinion on that. So this is a real life example from a mobile laboratory operated by the NHLS in our province in a peripheral setting. And it is a patient who had a negative rapid antigen test result, which says it does not exclude the possibility of SARS-CoV-2 infection. PCR testing is advised for persons who have compatible clinical symptoms or who have been in contact with a positive case. And that, I think, expresses it very nicely. It does have a value and a role to play, but one has to be very careful and really qualify what it does, what it can do, what it can't. Of course, there is a huge number of rapid antigen tests that are available, and at some stage, I was flooded with adverts a few times a day. The problem is that you need to make sure that the quality is good. Luckily, there are organizations like Find that really devote themselves to looking, and I just see that the link on the top one is incorrect. We'll correct that afterwards. Diagnostics Global Health is the other link that collect validations and evaluations for these tests and update that on a regular basis. So this is where I would look. And then, of course, there will be, depending on the national setup, there will be regulatory authorities in many countries. Um, and this is now, so those two links were sort of misplaced. Diagnostics Global Health is one of the sites where you can look up validations and evaluations for rapid antigen tests. And there's a training package available from Find, which is also useful if you want to introduce them, for example, for community settings. Now let's move to something that we all are going to face in diagnostic virology more and more as the pandemic progresses. We have a patient now who is known to have had COVID-19 before and had a positive lab test at the time and now test positive again. Now the question is it recurrence? Is it basically the same virus that they had before? Have they become reinfected with a different strain of virus? Or is it just residual viral RNA being shed? So it doesn't actually mean active infection. It's just residual shedding of RNA and no longer infectious. And that has implications for isolation, quarantine, and other things. And there are many confounders in terms of differences between patients and tests. So how to handle these cases? And I think that's something we all need to grapple with, and we're doing that on a daily basis. Prolonged viral RNA shedding is known to be very common. It does not reflect infectious virus. It does not seem to be linked to persistence of clinical illness or so-called long haul or long COVID. And it can lead to positive and negative results alternating if the viral load is low, and sometimes it's detected, sometimes not. So what we recommend is do a careful clinical and epidemiological and lab review, looking at illness, indications for retesting, exposure, and then also at the lab results. And here the CT values of real-time PCR tests can give an indication of the amount of virus present and can help to decide whether the second episode is actually just residual RNA shedding. So this is our own criteria for investigating possible reinfections if both tests are strong positive, are a month or longer apart. We then try to obtain all available samples, which is a problem because many labs don't store them for any period of time. Retest to confirm, try and do sequencing from both samples to confirm that there are different viruses. There will be subtle differences, but they should not be identical if they are indeed due to reinfection, and then ultimately one can confirm that they're both from the same patient. What we have discovered is that for all these new tests, we really need specimen banks and biorepositories. And that is not easy, again, much more difficult than with serological testing, where it's just a serum sample. Different types of samples from different body sites. One needs to document the patients and their characteristics very well, but they are important. And last but not least, all routinely tested samples should ideally also be retained in storage, i.e., frozen, for at least a month, if possible longer, to investigate outbreaks, which will take a few weeks to realize and then go back and sequence. And if we want to investigate reinfections, we would need to store samples for months to years. And as importantly, we need to evaluate these new tests. So having well-characterized banks of specimens available will be invaluable for that purpose. These are the priority areas, and I think we've covered this. So I'm going through this. I'm just going to skip it now. I just want to home in on two very special issues. The first is molecular epidemiology. And here I just want to draw your attention to some study that's now being submitted to a scientific journal on the molecular epidemiology of the early SARS-CoV-2 infection cases in Cape Town. And using sequencing, it was possible to show that Cape Town received seeding of our epidemic back in starting in March from various countries, particularly several European countries. And that is, of course, very important if one wants to work out the spread of the virus and try and control it. And the second thing is something we are currently doing, wastewater epidemiology. One can use viral RNA in sewage as a marker of epidemiological activity in a population. That is being done routinely now in Cape Town, where water coming into wastewater treatment works is routinely tested and also quantified. And we have seen, so we knew what was in store for us. We saw since November a rise in viral loads in sewage, and we knew that there were more infections occurring in the population. So the current increase in hospitalizations and also COVID-related deaths is not unexpected. And what we are now trying out is whether the same approach can be used for certain institutions, typically places where people reside. So that would be campuses of universities where people live in dorms. It could be prisons, it could be old age homes, where one can have an early warning system. Ideally, one would hope not to detect any viral RNA in the sewage, but if it is there, one can then monitor its development and possibly intervene in time by mass screening and then isolating the infected cases. Before I end, and this is my last slide, I would just like to draw your attention to this free reference book. It's again, it's something that is freely available online under this URL. It doesn't cost anything, and it is constantly updated. With this, I would like to thank you very much and hand over. Thank you.
Thank you so much, Wolfgang, for the thorough review of both PCR and antigen testing. I know there are many questions that are accumulating in the chat and that we will address in the question and answer service to follow. For now, I'd like to move on to our second esteemed speaker. Dr. Wilbur Sabiti is a biomedical research scientist and senior research fellow in medicine at the University of St. Andrews. He leads translational research on biomarkers for diagnosis and treatment with particular focus on respiratory diseases. Together with his colleague, Stephen Gillespie, he is spearheading the translation of novel tuberculosis molecular bacterial load assays for rapid monitoring of TB treatment and response. Like many of us, however, he has been pulled into the COVID response and will be speaking to us today on serological assays and their use in the pandemic. Wilbur.
Thank you, Chris. I'm Wilbur Sabit, and I come from the University of St. Andrews. University of St. Andrews is in the northeast of Scotland, 1 hour from Edinburgh, and we are known for being the home of golf and having educated the next in line to be the king of the United Kingdom, Prince William. So we're going to talk about serological testing, building on what my colleague Wolfgang has shared on molecular and antigen tests. So serological testing provides a supplementary diagnostic tool to molecular tests. The serological tests can be used for point of care testing and provide faster results and are less costly compared to molecular tests. Serological testing could be for population level screening or diagnostic support. Rapid development of serological assays for COVID-19 has been witnessed as part of the pandemic response. However, discrepancies exist between claimed and actual performance data for serological tests on the market. So manufacturers have come up with so many tests. The enthusiasm for developing tests has been so big, and we're receiving many claims of 90%, 100% sensitivity and specificity, but we're not sure whether the claims are matched with reality. So this is just a summary of how the human immune response comes about, especially for COVID-19. There are three major immunoglobulins that have been a target for COVID-19 serological testing, i.e. IgM, IgA, and IgG. IgM antibodies are produced during the early stages of the infection, and IgG is often the most abundant and plays a significant role in later stages of infection. So there's a late kind of onset of IgG antibodies. So when the host cell is invaded by the virus, the current understanding is that it binds the angiotensin receptors using its spike protein, and the entry into the cell kind of induces a cascade of immune responses, especially the antigen presenting cells, which presented to other immune cells like T helper cells, and this results in the production of antibodies. And as we know, IgM antibodies are the first to be produced, and then later on we have IgG coming into action. So there are currently four major types of serological diagnostic tests. So we have what we call rapid diagnostic tests. These take about 10 to 30 minutes, and they detect presence or absence of antibodies. So they are qualitative in nature. They don't give you a quantitative value of the result. Then the second group is the enzyme-linked immunosorbent assay, commonly known as ELISA. This takes around 2 to 5 hours. It takes presence or absence of antibodies, and it is quantitative in nature. Then the chemiluminescence immunoassay, it takes around 1 to 2 hours. It's quite faster than the ELISA. And it also detects the presence or absence of antibodies against virus present in the patient serum. Then we have the neutralization assay. This takes around 3 to 5 days to get a result. And it detects the presence of active antibodies in the patient serum that are able to inhibit virus growth ex vivo in cell culture system. So this is really looking for protective immunity, because it's one thing to have antibody response or an immune response against COVID, but it's another thing to have a protective immune response. And the neutralization assay looks for the antibodies that can really inhibit the virus. Then there's another type which has recently been developed. It's called the microsphere-based antibody assay. We can also define the serological test by type. Of course, the antibody test, which tells presence of antibody in human blood, and the antigen test, which Wolfgang has already talked about. This is whereby antibodies are the ones embedded in the lateral flow strip, and the antigen, either the virus from the patient sample, is added to the lateral flow. And if the patient has the antigen, then we'll get a positive result. So this is a diagrammatic summary of the different platforms, serological platforms. So the lateral flow test, or the rapid diagnostic test, it has a small port where the sample is added with buffer, and then it moves through the strip by lateral flow, and at some point, antibodies, the antigen is embedded within that part of the strip, and if the blood of the patient or any other suspected case has the antibodies, then a reaction will occur and we can see a positive result. So, and then we have the ELISA, where the antigen is bound on the plate, and then blood is added, blood sample, the neutralization assay where we see the viral prex and so on. So I want us to, at this stage, to have a poll to see where can we apply these serological tests. So the question is, in your opinion, what is the best application for COVID-19 serological tests? Primary diagnosis, vaccination response, serological prevalence, all the above or none of the above? I'll give you a few seconds to answer, and after that we shall look at the answers. Meanwhile, we'll keep talking about the different types. As I said, the neutralization assay is whereby the serum is added to the sample that has virus in it, and then to check whether the serum has antibodies that inhibit the viral growth. So we can now see what the responses are for the question. So very few think they are good for primary diagnosis, and I think I agree with them that they are not good for primary diagnosis. 14% I think it could be used for vaccination response. Seroprevalence, 47% seems to be the highest number of where they think we could use these antibodies, and 30% think we could use them for all of above, and 2% said none of the above. Thank you very much for your responses. That give us an idea of how people think about these tests. So timeline to antibody response in COVID-19. The antibody response really appears a bit later after the viral shedding. Day 7 seroconversion appears in 50%. So meaning that the majority, the seroconversion comes around 13, 14, and 13, 14 days following viral shedding. So IgM emerges quite early, around 4 days. And so that means that antibody tests are more useful in detecting previous SARS-CoV-2 infection if used 15 days or more from onset of symptoms. So the performance of serological tests, many serological diagnostics have been developed. And now, according to the databases on Find Diagnostics, 241 rapid diagnostic tests have been developed, and 159 manual stroke automated immunoassays, and 146 have been commercialized, while 222 rapid tests have been commercialized. Developers have provided different performance claims. This calls for robust, independent post-market evaluations. So unfortunately, very few have been evaluated in Africa. Performance depends on serological testing method used. A systematic review showed that pooled sensitivity was, for ELISA, 84%, lateral flow, 6%, and chemoluminescence, 97%. So this is kind of a diagrammatic representation of how the specificity and sensitivity works out for these serological tests. So for IgM, the specificity is quite high, early on in during infection, but sensitivity is very low. Then for other immunoglobulins like IgG and IgM, specificity increases as time goes on during infection, and also sensitivity increases as the time of infection increases. So the timing is very crucial for these tests, because if they are not used at the right time, then they may not give the right answer that is useful to the clinician or to the patient. So the utility of serological tests in Africa really is that recommendation from Africa CDC is that we should choose tests with high performance characteristics confirmed by an independent evaluation using large sample size. So that's something that we should look for when we are procuring these serological tests. So at present, use of antibody-based RDTs can be considered in the following situations. Triaging symptomatic individuals in healthcare or community settings, testing of contacts of confirmed COVID-19 cases, or informing situation analysis and surveillance. So as we mentioned before, when should someone go for this the serological test? So timing is crucial because viral shedding and antibody response may take days to be detectable. So 5 to 8 days for PCR following suspicious contact. That's recommended by NICD South Africa. Expensive, but the PCRs are expensive and the turnaround time is 24 hours. And then over 10 days following infection, antibody tests can be useful. For example, IgM can potentially be detected earlier than this. So they are simple, cheap, and they are positive. The positive result does not mean active infection. That's something that we need to note about serological tests. And the negative result does not mean no infection. So a little bit of puzzle on these serological tests. So lifespan of the antibody response post-infection is still a question mark, and also an issue that vaccination will have to grapple with. As the vaccines, we are going to get able to generate long-term protective immune response, or it's something that will disappear quite soon. So the way forward, so to be useful in Africa for serological tests should be sensitive and specific. They should be clinically meaningful, and because if something is not clinically meaningful, really, the test might be really a waste of money. Cheap and able to scale up. So unfortunately, Africa and Asia have only tested 1% of their populations compared to 10 to 20% in Europe and USA. So we can also apply these serological tests for seroprevalence studies to understand the extent of infection. Unfortunately, I've been looking around. I haven't seen many seroprevalence studies done in Africa. I think one in Kenya, just in blood donation samples. I think some studies going on in South Africa, but I haven't really seen really many nationwide seroprevalence surveys, which I think would be important to give us the state of this infection in Africa, because we know that the extent of PCR testing has been very low in Africa. So in order to have an idea of COVID-19 really burden, these serological tests could help in doing a quick, rapid seroprevalence. And it could be used to evaluate effectiveness of the forthcoming anti-COVID-19 vaccination. So they can also be used for confirming infection in individuals who have clinical illness that is suggestive of COVID-19, but who have received negative PCR result. They can also be used in identifying individuals who have been exposed to SARS-CoV-2, who can donate their blood to be used as a possible treatment for critically ill people with COVID-19. We know that plasma from people who have previously suffered from COVID-19 has been found to be quite effective in as a therapy to other patients. They can be used in vaccine trials to determine effects of experimental vaccines. Testing for the antibody enables researchers to see which participants have been exposed to the virus. In determining the extent of the epidemic, for example, they can be used to investigate outbreaks of COVID-19 in communities or in healthcare workers or other workplaces and or schools. The question is, why is uptake still low and how can it be accelerated? Particularly in Africa, the uptake of serological tests has been quite low. What can we do to accelerate the uptake of serological tests and to do more seroprevalence studies? That's the end of my slides. I want to acknowledge Dr. Winters Mutamba for his support in putting together the slides and the funders who fund my research at the University of St. Andrews. Thank you.
Thank you so much, Wilbur, for that excellent review of serological assays. So I know that there are many questions in the chat, and we will move into the Q&A session. Professor Shaima Inani will be leading that session. She is my co-chair in the Virology, Immunology, and Diagnostics Working Group and an associate professor of microbiology at the Suez Canal University in Egypt. She received her PhD from the School of Medical Sciences in Japan at Nagata University and did her postdoctoral studies in both the United States and in Japan. Professor Inani is a member of the COVID-19 diagnostics group in Africa. Dr. Inani, please lead us through the Q&A.
Okay, hello. Thanks, Chris, for the introduction. We have today two interesting talks and we have many questions. So let's start with the first question to Dr. Fraser. How reliable are CT values from PCR tests to give an idea of viral load in the end?
Excellent question. And I have to say, all of them are actually very good questions. There are good studies that correlate viral load values. And again, keep in mind the CT values, of course, depend not only on the test used, so they are not standardized in any way. They also depend on the quality of the specimen, which is also very difficult, if not impossible, to control. But they are, roughly, I would say that most studies would point to a CT value of around 30, indicating, so anything 30 and below would likely reflect infectious virus that could be isolated by cell culture, which is again not a standardized method. And anything above 30 is likely not to be infectious. It's very rough, but I think that's the best we have at this stage.
Okay, thanks, Professor. The second question is, can we use sputum to identify COVID and what is the sensitivity?
Yes, thank you. It can be used. Not all tests are able to use it. So that is something worth checking. And of course, if a test doesn't list this as a potential type of sample, doesn't mean it won't work, but it has to be validated by the lab. We do not get a lot of sputum samples, I have to say. So I'm not sure how important it is in practice, but for people who are, for example, who can produce sputum, it can be done.
Okay. The next question, I think maybe for both of you, Wilbur and Wolfgang, what are the best tests to use for screening a group?
I think that is really an overarching question that we both need to answer, because it really depends on what one wants to find. So acute infection, of course, would be PCR. Again, rapid antigen testing may be good enough for certain epidemiological purposes, for surveillance purposes. But then if you want to look longer term, it would have to be serosurveillance. Not sure whether Wilbur wants to come in here.
Yeah, I agree with Wolf on this, because as we mentioned in the presentation, antibody when it comes a little bit late. IgM comes early, but it's less sensitive in the early stages of infection. But maybe as a screening tool, yes, it could be used. But yeah, long term, serological test would be kind of useful and a cheap version for doing screening, because a PCR for screening is very expensive and not easy for many countries to pick that tab.
Okay, thank you. Another question, if someone continue testing real-time PCR positive even after a month, how do we make sure the person is non-infectious? Also, how can a virus become non-infectious?
Yeah, it's a good question. So basically, I'm a firm believer in timelines. I think we should not test people who have had a diagnosis of COVID. I think all we need to do is have a calendar. And I think there's very good evidence that for the vast majority of cases, with the exception perhaps of severely immunocompromised patients, after 10 days, the patient will stop shedding infectious virus, but may for much longer continue to shed viral RNA. The exact mechanisms I do not know, but I assume it has to do with quantities. There is a certain minimum quantity that would be needed for it to be infectious. So the viral RNA, and that is the beauty of real-time PCR, that one can pick that up by by a very high CT level, CT values, which basically reflect very low viral loads. And if I may, at the same time, deal with another of the questions, those samples are those that are often issued as equivocal, where we can't really make a call on whether that is true or not. However, if we know the history, and I know that's challenging because they may go to different labs and the clinical information may not be given, but if we know that somebody was positive before and we now pick up a high CT value, we know that this is real, but it's basically, it's on the way out. If, however, this is the first sample from a patient, we would not be able to tell whether this might be very early on in the case of infection.
Wilbur, would you like to add something?
I mean, someone who keeps testing positive for the virus, I think that kind of brings me back to what I mentioned in my presentation about the clinical meaningfulness of a test. So if someone is testing positive, but clinically they're not having any symptoms, or they are not really showing any severe disease, then they shouldn't be a cause for worry. So the clinical meaningfulness, I think, plays a part in decision making in that kind of a case.
Okay, thank you. Another question is, how stable is the viral RNA in the wastewater to be used for the API evaluation?
It's surprisingly stable. I think it has actually now opened up very rapidly and massively a whole new field, because wastewater epidemiology has historically been used, of course, for viruses transmitted in the fecal-oral route. Poliovirus being the most notable one, where many countries have surveillance systems to pick up, wild type or vaccine-derived poliovirus. And these are non-enveloped viruses that we know are very stable in environment. So to now have an envelope virus which is shed in sufficient quantities by a sufficient proportion of infected people to be picked up in the wastewater, and I suppose a lot of it actually comes from the respiratory tract, but then gets swallowed and passes through the digestive tract, and then survives, not in infectious form, but with sufficiently intact RNA for detection by PCR, and that it then survives for days to be picked up is I find quite remarkable, and I wonder whether going forward we might actually expand that to to test also for influenza activity and others.
Okay. Sharma also asking if can you please share some understanding on RNA shedding? What does it mean and why does it occur?
Yeah, I don't understand it well, I have to say. It may well be something that one finds
Maybe
more
she
often.
wants to clarify something about the RNA shedding.
Yeah, it is basically the phenomenon that people will be positive by PCR with increasing levels of CT values for prolonged periods of time in some cases. I don't understand it well, but I wonder whether it is actually rare or whether it is something that we are just noticing here because we are so nervous. Remember that early guidance from industrialized countries demanded 2 consecutive negative PCR tests on anybody who had COVID before they could leave isolation, which we now realize, and they have walked away from that, is not necessary. We can just count the days in the calendar, and I'm very confident that 10 days after symptom onset, whatever the PCR says, and I would prefer not to waste a precious PCR test on such a patient, whatever it says, I'm very confident that this patient will not be infectious, or at least not be highly infectious. So the usual precautions that we should all be maintaining, mask distancing and so on, would completely suffice.
Okay, thank you. And also we still have many questions. So what about cross-reactivity with other coronavirus in RDT?
Sham,
Okay.
Wilbur, over to you.
go ahead, Wolf, first.
Okay, because I think we run off time. Maybe
We run out of time. Yeah, it's a.
Let me let me kind of put a wrap on it. Cross-reactivity we know varies across the different assays, and we're learning a lot more. We've actually done a number of those studies in our group. And on that note, I will say thank you to our audience for your participation, for your questions. Say thank you to those who have led the webinar, Drs. Pricer, Sabiti, and Inani. Thank you all very much. This is to be the first of several webinars from our working group, so I hope you will join us in the future. The webinar recording will be published on the Coalition's website soon, and you will receive an email about a brief survey. We do hope you'll take the time to give us some feedback, and it'll be much appreciated. Again, if you're interested in joining our working group, please do not hesitate to apply via the chat that we have here, and we'll continue to try to answer some of those questions on the chat and otherwise, or address in later webinars. Thank you very much for joining us today.